Good morning, everyone. Shall we start? Are we ready?
Yes, sir.
Okay. Welcome, Dr. Ravi. Thank you for, first of all, accepting to do this particular webinar for the benefit of our students, faculty, staff, and also some of the well-wishers and friends of Indian Academy Group of Institutions. This initiative has come from the principals of our Indian Academy Degree College, as well as our School of Management Studies, Indian Academy Pre-University College, and Indian Academy College of Nursing. Requested me to organize this particular interactive exchange, knowing about you and seeing your presentations elsewhere in the media and the print media as well as electronic media. They have requested me, and I should thank you for readily accepting our invitation and joining us. Before we start, and I would like to take a couple of minutes to briefly introduce Dr. Ravi to our audience who have joined us. Dr. Ravi doesn't like to be a very, a fancy introduction, always wants to be a one-line introduction, but just let me give a few details about Dr. Ravi. He did his MBBS from Madras Medical College and MD in microbiology from Jipmer Pondicherry. He's also fellow of Indian Academy of Sciences and fellow of American Society of Tropical Medicine and Hygiene. Started the Department of Neurovirology at NIMHANS back in 1985. And I must say that I knew him from since then, almost, as we have been associated in NIMHANS in our initial years. While I was pursuing my PhD, Dr. Ravi just then joined. And Dr. Ravi is well known as one of the best virologists, scientists that our country has produced. He has published many research papers in both in national and international journals, and he is a recipient of 25 research grants at national level from granting agencies like ICMR, DBT, Department of Science and Technology, CSIR. And at international level, is a recipient of research grants from Indo-USA Vaccine Action Program, NIH and CDC USA, MRC, and University of Liverpool and National Institute of Health Research, United Kingdom. Some of these funding agencies have funded his research grants over a period of time. He is also a recipient of WHO Fellowship in 1992, Commonwealth Medical Fellowship UK in 1995-96, WHO Short-Term Professional Zero in 2006-07, International Examiner for MD Virology Program of MRI Sri Lanka since 2007. Dr. Ravi is a visiting professor of microbiology and immunology, University of Liverpool, UK since 2013. As you already know, Dr. Ravi had conducted an interactive program in many mediums, and he is on various boards right now, helping state and central governments as an advisory level. Is also on the International Advisory Board of Sputnik Vaccine. These are some of the few, details to introduce our eminent and famous speaker, Dr. Ravi. Dr. Ravi, over to you. Primarily, just want to give you, in the context-wise, you will be addressing a large number of our undergrad and master students who have concerns about how this pandemic is going to, open up, and how it is going to impact their studies in the next couple of years who are in UG program. Some of them are in the final year. So they are more concerned about their academic career and how it is going to impact. They would like to see some kind of a positive things to manage the situation and move on. So at the same time, they would like to know about the precautions they need to take. They like to know about various misinformation that is flooding in different students are active in social media, but that is the media where there is a lot of misinformation also. So try to enlighten and help all our students and faculty how to cope with this unfolding pandemic. Thank you, and over to you, Dr. Ravi.
Thank you, Dr. Somasekhar. It's always a pleasure to come to the Indian Academy of Sciences, and thank you for your very kind introduction. It's also nice. I also want to acknowledge the presence of Dr. Sunil and all other officials of Indian Academy. Good morning, and what I will do in the next 35-40 minutes is probably talk to you about COVID-19 vaccines and variants, which Dr. Somasekhar told me it will be nice if we can give some insights, and probably we will discuss later on in the question answer session if you have any concerns. Let me start with a disclaimer. All information in this talk is based on scientific evidence from published literature. It's certainly not from WhatsApp University. And you can see, unfortunately, I was not awarded the Doctor of Medicine in WhatsApp University. I don't know if any of you have. If you do, please forgive me for not using information from WhatsApp University. I have no conflict of interest to declare. Definitely, I don't have shares in any of the vaccine companies. So I try to be as factual as possible, and I also refrain from being prescriptive because I think you have to choose what you want to do. I'm going to give you a brief introduction to the virus, the severe acute respiratory syndrome, coronavirus 2, SARS-CoV-2. Then talk about variants, especially the nomenclature, the emergence, and the implications of variants. Then also touch on what are the types of vaccines, their efficacy, and issues surrounding vaccines, and then end with a summary. We all know that coronaviruses have been around. They derive their name corona because under the electron microscopy, they are spherical, and on their surface, they are studded with a protein known as spike protein, because this gives the appearance of the crown of a king. So because of the way it looks under the microscope, electron microscope, it was called corona to describe its shape. They are viruses that measure 60 to 120 nanometers. They are RNA viruses. You all know that viruses can either have RNA as genetic material or DNA as genetic material, unlike all other living species which have both RNA and DNA. We have known about 6 coronaviruses prior to the emergence of this new one. 4 of them have been around for 50 years. They cause mild infections in human beings. 229E, OC43, NL63, and HKU1 are very mild coronaviruses. They all cause either common cold or mild diarrhea. Turn of this century, in 2002, the first severe acute respiratory syndrome virus emerged in Guangdong province of southern China. And then from there, it spread to many countries, very quick, mainly through air travel. Patients who got infected traveled to many continents, and there were outbreaks in those countries as well. Fortunately, India was spared. But very quickly, we contained SARS-CoV-1, as it is now called. In 2012, we had another coronavirus causing severe respiratory syndrome in the Middle East. It's called MERS, Middle Eastern Respiratory Syndrome Virus. It also spread to some countries, but fortunately it didn't come to India. Now, these viruses are widely distributed in nature. Not only all these, what I told you, are viruses in human beings, but there are animal coronaviruses. The coronaviruses of bats, there are coronaviruses of birds, and so they are present in a wide variety of living creatures on the planet. And very frequently, they can cross over from one species to another, and we call them spillover events. Now, all the three severe acute respiratory syndrome viruses, namely SARS-CoV-1, MERS, and SARS-CoV-2, the reservoir in nature is bats. From the bats, it has spilled over to human beings, and usually when spillover occurs, there's an intermediate host. In the case of SARS-CoV-1, it was the civet cat. In the case of MERS coronavirus, it was the camel. And in case of SARS-CoV-2, it is the pangolin, which is an anteater. Incidentally, bats and pangolins were sold in a crowded wet market of Wuhan, which is a province in southern India. The virus is spherical, as I mentioned, and it is about 60 to 120 nanometers in width. It has an outer envelope, which is shown on the left as a blue circle. This is a lipid envelope. Embedded in the lipid envelope are three proteins. The spike protein, which is the most important protein of the virus. It has a globular head and it has a stem, a linear stem. Now, this is the most important protein of the virus because using this protein, virus attaches to human cells. So that way, this is actually the life of the virus. This protein represents life of the virus. Second, all protection is directed against this protein. So it is a very antigenic protein and it induces a very potent immune response, and that immune response is protective. So both in terms of virus attachment and protection, this is one of the key proteins. Apart from that, we have membrane protein, nuclear protein embedded in the envelope. The nuclear protein surrounds the nucleic acid, which is RNA, and this RNA is positive stranded. So this is the three-dimensional structure of SARS-CoV-2 spike protein has been worked out very early last year. And as I said, it has a globular head and a stem. In the globular head, there is one portion of the spike protein that binds to the receptor. This is popularly known as RBD. RBD stands for receptor binding domain. And the receptor that this virus binds to in human cells is angiotensin converting enzyme 2. Now, this is a physiological molecule present on human cells to regulate blood pressure. So, angiotensin and renin hormones bind to this. So, these are basically meant for blood pressure control, but the virus attaches to that receptor. So this protein, especially the receptor binding domain, is very unique to SARS-CoV-2. It is not shared with SARS-CoV-1 or MERS coronavirus. So naturally, that makes it a very interesting candidate for developing vaccines. The genome itself is about 29,000 nucleotides long. It has it's a huge single open reading frame, meaning if you introduce this RNA into cells, immediately it can go into translation. And in the 5' end, you have the open reading frame, huge open reading frame called 1A and 1B, followed by all the proteins of the virus, the spike protein, the envelope protein, the matrix protein, and the nucleoprotein. So the spike protein is roughly almost in the center of the entire genome. Moving on to variants of SARS-CoV-2. Now, last year, around this time last year, we knew that coronavirus emerged in Wuhan, and by January 5th, the complete genome was sequenced, and the virus started moving out of China to other countries, and the mutation was moderate. It was not very, like influenza virus, it was not mutating very fast. One of the first mutations we saw in the coronavirus was detected in Italy. by end of February. And there was one particular amino acid in the spike protein at position 614, which changed from aspartic acid to glycine, and that made the virus more infectious. So we knew variants were starting emerging. And as variants started emerging, there was a lot of confusion on how to give names to them. There were 2-3 systems that were existing. One system was adopted from the global influenza database, which is the GISAID database, where all influenza genomes were being stored. This database was developed somewhere in 2006-7, and after the swine flu pandemic, all genomes were being deposited here. And then subsequently, the scientists in the world decided, let this be an open database to store all viral genomes. So when MERS came, those genomes were put into it. When Ebola came, those genomes were put into it. When Zika came, those genomes were put into it. So even for SARS-CoV-2, people decided that they will put it into gisaid.org. That's the website where all genome sequences are deposited. So they had their own nomenclature. But Nature Medicine, Nature Microbiology published a paper last year saying, for uniformity, let us have a single way we will call these variants. And that is shown here. So SARS-CoV-2 lineages and variants are designated with alphabets now. The parent Wuhan virus, which emerged, was called A. And if there were some nucleotide changes which did not amount to a great percentage, they were all in A, but they were called A1, A2, A3. But when there were some significant mutations, they gave it a second alphabet B. And B is the first mutation was detected in China, but B moved out very fast out of China. And in Italy, I told you one mutation occurred that was B.1, that is D614G. And subsequently, as the virus is infecting large number of people, it is mutating. And based on the sequence, it is assigned new lineages like B.1, B.1.1, B.1.17, and so on and so forth. So that's how the nomenclature we follow right now. This is the map of the SARS-CoV-2 genome. In the middle panel, you can see that we have the ORF proteins. The spike protein is colored red, and then you have the other proteins. And as of today, I think close to 20,000 mutations have occurred in SARS-CoV based on the first sequence that was released in January 5th, 2020. So the virus is constantly mutating, and many of these mutations are in the spike protein. As you can see here, too many red lines are there. The density is very high. And in the bottom, you can see the Manhattans also, the density is very high because they are dark there. Okay, so what is a variant? A variant is any virus nucleic acid where the nucleotides compared to the parent virus have changed, and mutations will continue to occur. We'll come to that, why it occurs in the subsequent slides. So we have to keep tracking the mutations in the virus, and we have to keep looking at emerging variants, because from the public health point of view, variants pose a number of challenges. One is you can get variants that can spread faster. That means increased transmissibility. We can get variants that can escape protection given by a vaccine. We call them immune escape variants. They will evade natural immunity. And we also, it's possible that we can get variants that can cause more severe disease and higher rate of death. So mortality, morbidity can be affected. We at present do not have specific antiviral drugs, but variants, if you take from the influenza perspective, we know that if there are antiviral drugs, because of mutation, the variants can escape even antiviral drugs. So variants are very, very important in any viral infection, and we need to keep tracking it. In the case of COVID-19, it is very important because, the number of infections that are being caused, it's a pandemic. And in our lifetime, we have not seen so many people getting infected. Therefore, you can imagine in one individual itself how many variants will be coming up. Variants are broadly classified as variants of interest or variants under investigation, VOI or VUI, when you start seeing it spreading to larger geographical areas. It may start with one state in a country, then slowly it can spread to other states in the country. If it spreads very fast across the globe, then it is called a variant of concern. And to call a lineage as variant of concern, you should have two important pieces of data. A, that the infectivity in people is more, that means the virus spreads faster than the parent virus. B, in cell culture, it should grow faster and to higher levels compared to the parent virus. If these two conditions are fulfilled, then it's called variant of concern. Why do variants emerge? Well, RNA viruses are endowed with the inherent advantage that they mutate. Why do they mutate? Because they have an enzyme known as RNA dependent RNA polymerase. The duty of this enzyme is to copy the genetic material of the virus. And when it copies that, it fails to proofread the new strand that is synthesized. It has poor proofreading ability. So mistakes creep in, and these mistakes result in mutations. Mutation is nothing but change in nucleotides. And when these mutations accumulate, they can change the protein for which they code. And if they change critical amino acids that change the charge in the protein, then virus behavior will alter. So inherently, RNA viruses, be it influenza, HIV, or SARS-CoV, or for that matter, other RNA viruses, respiratory viruses, they all continuously keep mutating and new variants keep emerging. The virus is constantly under evolution. The second reason why viruses mutate is the immune system puts pressure on the virus. When the virus enters a host, it multiplies, immune system develop antibodies, they try to neutralize the virus, and the virus in response mutates, trying to escape the neutralization. So, immune response also is responsible for viruses to mutate. In fact, one of the theories why the UK variant has emerged was it emerged probably from a person who was immunosuppressed, who had received plasma therapy, and who had a very chronic infection with SARS-CoV-2. So in his body, probably it mutated. What are the implications of variants? Why am I talking so much to you about variants? Why should I, labor you with so much of information? Variants can alter transmission, they can alter replication, and most importantly, from today's perspective, they can escape vaccines. So let us see what is the status. Now, one of the first variants of concern, that means one of the variants that spread globally, replacing existing viruses, was detected in the United Kingdom, and it's popularly known as UK variant. Now, this is a map of the UK variant. It was detected in UK late last year in November, December, early December. And very soon, with international travel, it started spreading to many countries across the globe. In the bottom, you see a heat map that is time related. In 3 months, you can see that many countries started becoming dark gray or dark green. Darker means more number of cases, lighter means less number of cases. So, in a matter of 3 months, it had completely uh, started replacing existing lineages in several countries. So this at that time was declared as a variant of concern. Today we have 5 variants of concern. One is the UK variant. On the right in the top you can see the name given, the nomenclature is B.1.1.17. So this is UK variant. The next variant that emerged was from South Africa. And that has all the mutations that the UK variant has, plus it has an additional mutation. It was detected in South Africa. Next was the P.1 variant that was detected in Brazil. Late last year in USA, in California, there was one that caused large outbreaks in California state, and this is called the B.1.427. The last variant of concern was detected in India. This also towards the end of December, but by the time we noticed it was February. But when we went back, we knew December samples had this. Now, this is a mutation over and above the Californian variant. Now, this very recently, 617, was classified as variant of concern a week back by Public Health England and also by WHO, because they have sufficient information in England saying that this virus is causing more infections compared to the normally other circulating virus infections. So it is more infections, 40 to 60% more infectious. And we also have cell culture data to say that it replicates aggressively. So the way all these variants have been found in India. We have UK variant, we have South Africa variant, we have Brazil variant, and we have, of course, variants detected in our own country since December. So all these are now circulating in India. This is a map from GISAID website telling you how the virus that was detected in India has moved. Today, it has moved to almost 50 countries. Last when I saw the GSAID website 3 days back, it was 48 countries. I'm sure another 2 countries would be added now. And these are the numbers and percentages. These are the top 15, 20 countries where you can see a large number of double mutant described from India is being detected. Coming to our own state, India has started sequencing sometime in late December. Once the UK announced that it had a variant of concern, it alerted the globe. Indian government swung into action and said we should now start looking for variants. So we should test all samples coming, all passengers coming from UK, and if they're positive, then we should start sequencing. So how do we go about? So they established what is known as INSA-CoV, Indian SARS-CoV Consortium for Genomics. INSA-CoV is the acronym. Now this consortium consists of 10 laboratories, research and hospital laboratories across the country. They are coordinated by National Center for Disease Control. It is funded by Department of Biotechnology, and their job is to do sequencing. And what kind of sequencing should they do? One, all international travelers who are coming, they're positive, because we'll have to be vigilant of new variants entering our country. So that is one group we will test. Apart from that, locally in every area which is mapped to a lab, if there are new clusters or super spreading events that occur, then from those clusters, some samples have to be collected to see it is being caused by a new variant. Second, from the community, if we have any severe form of disease in groups, then to ask the question, is it because of a variant? We collect samples. Younger age group, especially children, we collect samples to know if there is a mutant circulating in children as compared to adults. And lastly, we collect samples from people who have received 2 doses of vaccine and have completed 2 to 3 weeks. Because in fully immunized people, if you are getting infections, then we'll have to ask the question, is the virus escaping neutralization? To detect neutralization escape variants, we need samples from that group. So we are not doing sequencing in every sample that comes because it's a very, very costly, laborious exercise. It takes a week to 10 days to get sequencing report. And it costs anywhere between 8,000 to 20,000 rupees, depending on how many samples you're doing at a time. This is the latest report from Karnataka. So far, between NIMHANS, which is an INSACOG lab, and NCBS, which is also in Bangalore, National Center of Biological Sciences, we have so far sequenced 933 samples, and we have detected 52 different lineages. But the most important ones are the UK lineage. You can see the panel on the right in the table below, B.1.17, that accounts for about 11 to 12%. Then we have the South African variant we have detected in 6 samples. The double mutant Maharashtra, which is called B.1.617, and there are 2 groups there, 6171 and 6172. 6171 was detected in Karnataka towards the end of March, and it started spreading fast. But then came 6172 in the middle of April, and that is completely taken over. And that is a predominant lineage, and that is shown in the graph here. Prior to January of this year, we had a variant called B.1.36.29, which was dominating in Karnataka. It is not a variant of concern, but it was the most common variant being detected. Then in February, we got 61.7.1. That started taking over. These brown bars you can see, the number of variants are increasing, and the green and the violet has already shrunk. But then you see in the last 3 weeks or 2 weeks comes the 617.2, which is actually called variant of concern. It is completely taken over, and right now almost all the sequences that we are getting from samples are 617.2. So this is dominating circulation now. The same is true of Bengal. This is presented in a wave form of a map. You can see in January, the red line here was very, very small proportion, but by April end, it had completely taken over. Same is true of all other South Indian states, where you can see this indigo bar is ever expanding, and it's now 61.7. Okay. What are the other issues or implications related to
Dr. Ravi, just to interrupt you for a minute.
Yes.
If you don't, yeah. Just wanted to inform all the participants. Some of them are raising hands, probably have a question. I forgot to inform audience in the beginning. The format is Dr. Ravi is going to complete his presentation in the first 45 minutes or so, 50 minutes. And later he has sufficient time to answer all your queries. Questions will not be taken during the presentation. And also, all audiences, audience are requested to put your questions in the chat box, and we will read out the questions to Dr. Ravi, and he will clarify your doubts. Go ahead, Dr. Ravi. Sorry for that interruption.
Thank you, Dr. Somasekhar. We already saw that transmissibility is one of the major implications of variants, and we have 5 variants of concern now. The second question people are looking with respect to variants is, does it alter the virulence of the virus? Which means, does it make the virus more deadly, cause more severe diseases? This is data evolving. Now that the UK variant, we have data saying that it was more lethal in experimental animals and also probably in human beings. Certain subsets of people are getting severe disease. As far as the Indian variant is concerned, it is second last from below, B.1.617. It is under investigation. The National Institute of Virology has submitted a paper where it has shown that this particular variant is producing more severe disease in hamsters, which is an experimental model. So right now it is under investigation. So like this, on virulence, there are some studies going on. As far as antigenicity goes, yes, people have started looking at the efficacy of vaccines with respect to variants, and I will not talk about it now because when I talk about vaccines, I will give you a small update on it. In terms of the multiplication in cell culture, you can see that the 617 has a mutation at 452 in spike protein from a lysine to an arginine, change in amino acid, and this has made the virus more infectious in cell culture. How do we say that? You see the graphs below in the panel, there are different graphs. The y-axis is the percentage of binding of these viruses, and the x-axis is the level of the virus. So the parent Wuhan virus is represented as a solid black line. So viruses that shift to the left means they are able to multiply much, much better compared to the Wuhan virus. L452R is there both in Californian mutant as well as the mutant identified in India. N501Y is the UK variant. So these experiments are telling you, even in cell culture, these viruses are able to multiply much better than the parent virus. Now here is the information on the vaccine. What is the implications for vaccines? Now these are vaccines listed here row wise: Johnson Johnson, Pfizer, Moderna, Sputnik, Astrazeneca, the Novavax, the Sinovac is the Chinese killed vaccine. Now people have collected blood samples from subjects who got vaccinated with each of these vaccines and then done neutralization test with the various variants. You can see the last column I boxed in red for you. As far as the AstraZeneca vaccine and the Johnson Johnson vaccine goes, it is only able to partially neutralize the UK variant. And if you look at the AstraZeneca, the neutralization is much, much less, only 22% it is able to neutralize. The Novavax, it's 49%. So the South African variant is notorious to escape many vaccines. But although it is not really escapes, I must say that it is neutralized to a much lesser extent. It is still being neutralized. It is still being neutralized, but it is neutralized to a much less extent. We'll come to that a little later when I talk about vaccines. What about our own Indigenous Co-Vaccine? Now, this vaccine developed by Bharat Biotech from a virus that was isolated by NIV. It's a whole virus killed vaccine. Now, this vaccine, people have been immunized and volunteers' blood samples have been taken and they have been neutralized with the UK strain as well as the local double mutant 617 or the Maharashtrian mutant as it is called. And the good news is it is able to neutralize equally well all variants. So this is a paper that is submitted for publication in bioRxiv. It is available, people can see it. Now, before I move on to vaccines, where I will spend 10 minutes, I want to burst some myths. The myths are new strains have made the virus more deadly. As I showed you on virulence, data is evolving. Right now, there is no evidence to say it is more deadly. There is no difference in mortality rate compared to the first wave to the second wave. In the first wave, these variants were not there. So, definitely, it is not more deadly. More people are dying because there are more number of infections, and because there are no beds, they're coming late to the hospital. One of the commonest causes for mortality increase is coming late to hospital. Now, this is a virus that is attacking younger people, but not older people. And this is again a myth. If you look at the age breakup of those who are infected in the second wave, all age groups are infected, starting from children to adults. Younger people between 20 to 45 age represent a large proportion in the population. India is a young country. So naturally you will have more numbers there. But if you see in terms of proportions, people tested and those positive, there is no difference in the age groups. The last myth I want to burst about variants is there's a lot of WhatsApp University news saying that the new tests are not detecting variants. This is absolute rubbish. Almost all tests we use, the PCR ones nowadays, are based on amplification of multiple genes. They're called multiplex PCRs. And we amplify more than one gene. Most mutations are in the spike protein. And in all the other genes where we are amplifying, there have been no mutations found. Consequently, the PCR is working absolutely fine. But the PCR cannot tell you if it's a variant or not. It will only show a positive result. But for variant detection, you have to use sequencing. Moving on to vaccines. Now, SARS-CoV-2 is one virus that has completely revolutionized vaccine production for several reasons. But the first reason is in 9 months we had a vaccine. From the time the sequence of the virus was known, which is phenomenal. In medical history, we never had like this. I want to introduce vaccines to you from the point of view of a virus. We already saw that virus is spherical. On its surface, it has spike protein, and inside it has an RNA. With respect to SARS-CoV, different types of vaccines have been formed. The conventional old-time methodology of making vaccines was to grow the virus, purify it, kill the virus using a chemical like either formalin or beta-propiolactone, and that's called inactivated whole virus vaccines. An example of that is the Bharat Biotech Co-Vaccine. But because science has advanced so much and molecular biology has advanced so much, we also have started taking shortcuts. One of the things is not to use the whole virus. We know spike protein is most important. So people have taken spike protein gene, inserted into other innocuous viruses like adenovirus, and made a recombinant viral vectored vaccine. Now, this vaccine carries spike protein on the surface, but the rest of the virus is adenovirus. And they have made it replication deficient, which means it can infect a cell, deliver the protein, and then die. And the immune system from then on will take on the spike protein and make antibodies. So there are two examples of viral vector vaccines licensed. One is the Covishield, made by Oxford University and Astrazeneca, produced in India by Serum Institute. And the other is the Russian vaccine, produced by Gamaleya Institute in Russia, and goes by the name Sputnik. Now, both are now in India. Sputnik has just arrived in India. Manufacturing is going to start for Sputnik, and in 3 months we'll have virus made in India, vaccine made in India. Covishield is being produced globally at a large scale by Serum Institute of India. For the first time in human history, people also developed nucleic acid based vaccines. We know the nucleic acid of SARS-CoV-2 is RNA, is an mRNA, and we know spike protein is the most immunogenic protein. So they took this mRNA for spike protein and then encapsulated it in liposomes, so that it can be delivered inside a cell. If you give naked RNA, it will be digested by the cellular enzymes. It will not enter cells. So you will have to hide the RNA in a lipid coat and then deliver it into the cell. The lipid fuses with cell. Now this is fantastic technology. It has been perfected in very short time. And I think this is it's one of the breakthroughs in science, because tomorrow we can start putting any molecule that we want to deliver into the cell. And this is a high efficient, transfer. So two mRNA vaccines, one made by Pfizer and the other made by Moderna, are made in America. In all these vaccines today, excepting one, I'll show you on the right side, and that is the Johnson Johnson. It's not there here. Are two-dose vaccines. That means you have to give two doses. The spacing between doses vary. For mRNA vaccines, it is 3 weeks apart. For Sputnik, it is 3 weeks apart. For AstraZeneca, when they tried it out, they tried 3 different time points: 0 and 4, 0 and 6, and 0 and 12. 3 groups of patients. And they found that the best response is between 0 and 12 weeks. But 0.4 also protects and 0.6 also protects. mRNA vaccines, as you can see on the table, have 95% efficacy, but they have to be stored at a very low temperature, which is a challenge for developing countries. Most of the other vaccines are stored 2 to 8. Sputnik is available as a powder, which can be stored in 2 to 8, but once you add and water and reconstitute, it has to go to -20. Co-vaccine, which is our own Indian vaccine, is 2 to 8 degrees. How do vaccines work? Before I tell you how do vaccines work, what happens in natural COVID infection? Natural COVID infection, your body responds. Within 2 weeks, you start making antibodies and your cell-mediated immune response mounts, and you get over the infection by 4 weeks to 6 weeks. 95% of the people have recovered. A small percentage do die. In those who recovered, the immunological memory stays for 3 to 5 months. Antibodies are found in high levels for 3 to 5 months. After that, the antibodies start declining. And by 6 months, most of them don't have antibodies. So the immunity in natural infection, we know over the last 1 year that it lasts about 6 months. So at the end of 6 months, many people started getting reinfections. They will get reinfections if it's with a new variant. It is possible, but they don't develop severe disease. Whereas vaccination will give you protection for 1 year. Why? Because in vaccination, you are deliberately giving a very high dose compared to the dose you receive in natural infection. In a short time, you are pumping the immune system with purified antigens of the virus that bombard your immune system and then make antibodies and cell-mediated immunities. Therefore, vaccine-induced immunity lasts slightly longer. How long it lasts? Right now, but because used, we believe from experience with other coronaviruses, it lasts for minimum of 1 year. At the end of 1 year, will it wane? The answer we do not know. We'll have to wait for the answers till we complete 1 year after vaccination. Let me burst one important myth and misconception that is there in many people, including many doctors as well. What is protection? Protection against disease and protection against death is what you get with vaccination. No vaccine gives you guarantee against infection. In fact, in the history of human medicine, almost all viral vaccines that have been produced and used only give you protection against disease and death, not against infection. Classical example, polio vaccine. Polio vaccine prevents paralytic illness. It does not prevent infection. influenza vaccine. It prevents severe disease. It does not prevent influenza. Measles vaccine prevents measles, but virus can enter your throat, multiply, and you can also transmit the virus to others. So please understand, you are getting protection against disease and death, but you are not going to get protection against infection. Now, this is very, very important, because even after vaccination, you have to wear mask and you have to behave responsibly, because you can get infected and you can transmit virus to others. So you have a moral responsibility. Good that you're protected, but make sure you don't spread infection to others. This is the other myth, is that vaccines have been made very fast. And these are experimental vaccines. I don't think they are safe. Let me tell you how vaccines are made. There are several phases through which a vaccine goes in development. The first is preclinical testing, where vaccines are initially tried in animals. The animal models, small animal models, are mice and ferrets, and the large animal is rhesus monkey. Unless these candidates are first tested, and these animals are challenged with virulent virus, and they should be protected, and they should not have any toxicity, only then it's cleared for human trial. Human trial is in 3 phases. First phase looks at safety of the vaccine, whether if you give it to about 20 to 100 volunteers, are they safe? Are they getting any metabolic abnormalities? If they are safe, then it goes to phase 2. In phase 2, we give it to about 100 to 200 volunteers. We ask the question, in human beings, when you inject, are you developing antibodies? Are you developing cellular response? So is the vaccine doing what it is supposed to do? And this is called immunogenicity. So phase 1, safety. Phase 2, immunogenicity. And phase 3 is efficacy. What is efficacy? Large number of people, volunteers, are given the vaccine, along with a proportion of people, usually 1/4 or 1/3, who receive some other innocuous substance like saline or vitamin injection. That's called placebo. Now, at the end of the 2 doses and 15 days later, you sample all these people to see what is the proportion of infections in placebo group and what is the proportion of infections in the vaccinated group, and then you decide whether it is effective or not. And depending on the percentage, you say it is 95% effective, means 95% of the people who receive the vaccine are unlikely to get infected. 5% can still get infected, like in the Pfizer and Moderna. Okay, vaccination is the most attractive possibility that human race has been pursuing, because actually what is much more simpler and less expensive is behavior change. Behavior change is wearing masks, maintain distance, washing hands. Now, if you ask human beings to do it for the rest of their lives, you can prevent a large number of infections, but they're not going to do it. So we'll have to rely on vaccination. It is acceptability is very high, and also it is expensive to vaccinate people. The whole process of vaccination in India has been devised in such a way, because it's a new vaccine, that there are various steps. First is registration, then verification of your registration with documents. Then you go to the site where this verification is done. It is to be given in healthcare facilities or anywhere where there is a doctor and there's adequate space, and you have emergency beds and emergency drugs in case somebody. It is not like polio drops, you can go door to door and give it. Then the vaccine is administered and you're asked to wait for 30 minutes, observation phase, at which time, if you have any reactions, the doctor and the nurse there can attend to it and treat you. So this is a process that will require at least 2 hours after you go to a healthcare facility. Remember that. So this takes time. It is not that if you have million doses today, you can give million people in Bangalore million doses today. No, we don't have the capacity to vaccinate million people in one city in one day. It will take minimum of 20 days or 30 days to give a million people. As of now, India has vaccinated more than 18.5 crore people. This was about 10 days back. This what you're seeing here. So it's about 18.5, 19 crore people we have vaccinated. Our population is 130 crore. And in this, many of them have received first dose, only half of them have received second dose. And if you look at the distribution of age, above 60 years, we opened vaccination first because they were most vulnerable. They were getting severe disease and death. So we gave it first to 60 people and healthcare workers, obviously, because they have to treat others. So this coverage is 40%. Although we started on January 16th, 4 months down the line, we still have covered only 40%. In the age group 45 to 60, which we opened up, we have covered 45% of people. And in the younger lot, most of you in Indian Academy Degree College, it has been opened up from first, and only 5% are covered. So coverage is evolving. The government has done two things. One thing is, because Covishield is being used for a large number of people, advise that people can take the second shot after 3 months instead of what it was originally 4 weeks, then move to 6 weeks. Now, this gives an advantage. In that 3 months, many more people can get one dose. UK adopted it first. It is a nice public health strategy. But does it make the vaccine less efficacious? No. There is enough science support from the original trial and subsequently from UK that delaying second dose of Covishield to 90 days will not compromise protection. And also the government has said, anyway, people who get infected are protected for 3 months. So those who have got infection should wait for 3 months before they get the vaccine. They should certainly get the vaccine, but not immediately, because they still have an active immune response. Now, those who have, for co-vaccine, it is still 4 weeks, because when the vaccine was cleared, it was cleared with data for 4 weeks. Maximum, you can wait another week or two. So up to 6 weeks, you can wait if there is shortage. Till recently, the vaccine was allowed only for adults between 18 years and above. And it was not permitted for pregnant women, lactating women, meaning mothers who are feeding babies, and for children. But right now, it has been released for lactating women because it has been found safe in many parts of the world. So India also said you can open up for lactating women. No screening of either antibody or PCR is required prior to vaccination. Even after vaccination with 2 doses or 1 dose, you can get infected. This is preliminary data from Indian Council of Medical Research with respect to Covishield and Co-Vaccine. There were so far, this was about 10 days back, 17,000 people who got infected after 1 dose of Covishield and 5,000 after 2 doses of Covishield. Co-vaccine, 4,208, but the number of co-vaccines administered is less. Percentage-wise, it is more or less the same, both after first dose and second dose. The point I'm trying to make is there will be people who will get infected. That's why adopting appropriate behavior is very important. There are several myths. I thought I'll burst them. It is not safe because it was developed very fast. The answer is no. mRNA vaccines get integrated into human genome. COVID spike protein mRNA does not get integrated into a genome on its own. The vaccine can give you COVID. No, we are not using live attenuated virus. We are using only killed virus in Covaxin. In all other vaccines, it's only spike protein or its gene. It contains egg protein. No, none of the COVID vaccines are made in eggs. It causes severe side effects. No, the common side effects are fever, soreness, pain in the site of inoculation, myalgia, headache. Some people have vomiting and diarrhea, but all this clears in 48 hours. Some people get severe side effects, yes, but that is always there with any vaccine. People will get anaphylaxis kind of reaction. Some people get neuroinflammatory syndromes, Guillain-Barré syndromes, clot formations, but the proportion is very, very small. And it's also very difficult to associate those complications with vaccine. There's a committee that decides that. Vaccines make women infertile. Now, this is anti-vaccine lobby campaigns. It has been there for virtually every vaccine that has been released for human use. This is not true. And also, there is a myth that women should not take vaccines when they have periods because it is dangerous. There's no danger. There's no compromise even to the immune response. So it is safe to take vaccine even during monthly periods. So I think these myths I wanted to break. Now, the biggest problem now is availability. Take India. We have 130 crore people. In that 30 crore are children, 100 crore adults. That means 200 crore doses. But even the entire globe is not making 200 crore doses. It will take time to make. And currently, if you look at manufacturing, it is 4.69 billion doses are coming from US. India is second. It's producing 3.13 billion doses, and other countries are making vaccines. Vaccine production, these are biological products. They are not pharmaceutical products that you can have a machine, make a powder, make them into tablet and put them into foil and send it out. You have to grow cells, or and then you have to, whether it's adenovirus vaccine or co-vaccine, you will have to grow the viruses, concentrate them, purify them, post-treat them, check their sterility, bottle them. Whole process takes a long time. Right now, India started ramping up production at Serum Institute and Bharat Biotech. It will take 6 to 8 weeks. So middle of next month or late next month, early July onwards, our supply chain will improve and we'll get more number of vaccines. We also are having very promising vaccines in the pipeline. I know of this because I'm co-chairing the DBT committee. And these are all very promising vaccines. Zydus is coming out for the first time with a DNA vaccine for SARS-CoV-2. But the problem is it has 3 doses, but still its trials are ending by this month end. And very interestingly, they have included 12 to 18-year-old children in this vaccine. We have a recombinant S protein being developed by biological humans, and that is also in advanced trials. Results will be out by June end. Genova is India's own mRNA vaccine, indigenous vaccine. This is in phase 1 trials now. We will have data by another 3 months. Bharat Biotech has produced a nasal vaccine. Experimental results are very encouraging in animals. This is in phase 1 trial, and we hope to get the results in another 2-3 months. And Serum Institute is producing a second vaccine, which is a HBsAg and SARS-CoV-2 spike protein, fusion protein. Now, this is also in phase 1 and phase 2 trials. So there are many vaccines under COVID Mission Suraksha, which the Prime Minister has created a fund of 1,000 crores. These vaccines are being produced. The target is by year end latest, large number of doses will be. And I also forgot to tell you, Sputnik has come to India, and Sputnik has tied up with 5 companies. Hopefully by December end, we'll have 800 million doses being produced in India. So that is the scenario on vaccine. Now, what about the way ahead? I always use this slide to tell the road ahead is a road that has many road humps, speed breakers. If you look at human history, whether it is Spanish flu or whether it is H1N1, all viruses that cause pandemics come in waves. Initially, a large number of people get infected, and when they get infected, they develop disease. And when they develop disease and people start dying, restrictions are imposed. Even during the 1918 Spanish flu, travel restrictions, isolations came into place, and also people realized this is something serious and start behaving responsibly. They stay indoors, they avoid crowds, they wear masks, and they practice hand hygiene. Then what happens? Wave subsides. Again, people get back to their routine. So in 3 to 4 months, again some small events, super spreading events, which I call as sparks. These sparks start flying in urban settings, in crowded settings. Many sparks light up a fire, like it happened for the second wave in India. There are many, many reasons for the second wave in India. People keep asking me. I think the first and foremost is all of us are responsible. We did not adopt COVID appropriated behavior like wearing masks, maintaining distance, avoiding crowding, and sanitization. Between December and February, everybody was happy, back to their routine pre COVID days, not following any precaution. On top of that, you had some parties, and if you take Bangalore, you had apartment complexes showing up more number of cases in parties, student hostels turning positive. Now, this also started spilling into the community. What was happening in the community? There were rallies, there were political rallies because of elections, there were religious congregations. All these crowds, the virus started thriving and slowly multiplying. So this led to a huge, huge surge. Much like what you're seeing in the middle on the right, a graph. The first wave was a small bump, but the second wave was very, very huge. Now, this is what is being seen not only in India. All European nations have finished second wave. Some of them finished third wave or in the third wave. America has finished first wave, second wave, third wave. It's in the fourth wave, in the declining phase. So there will be a third wave, whether you like it or not, for two reasons. You get immunity against SARS-CoV-2 either by infection, so you have to be infected, or you should be vaccinated. But more than half of India is neither infected nor vaccinated. So there are still people who are susceptible to viral infection. Believe me, viruses will seek out susceptible people and cause infection. Generally, waves will be spaced 3 to 5 months. So the predictions currently of some of the people in the field, including me, is that we'll have a second wave towards the end of the year, third wave towards the end of this year. Because it's a new virus, you get immunity only either infection or vaccination. We are not going to complete vaccinating 100 crore adults by December. That's a pipe dream and it's not going to happen. Children are going to be highly susceptible. Why? Because in the first wave, 4% got infected. In the second wave, you can see data from Times of India yesterday. We have a large number of people infected in the second wave. You can see 1,44,000 people have been infected between March and May 18th in Karnataka, compared to 93,000 prior to March, entire period, March to March, 1 year, only 93. So infections are spreading in children. We don't have a vaccine for children. So there will be a large pool. India has 30 crore children. So in the next wave, the virus will definitely seek out children, and you should expect a large number of infections in children. What should we be doing? There is no alternative. We have to wear masks. We have to maintain social distance. I think we have to accept it. All of us have a responsibility, not only of following it, but also ensuring others follow it. And even our social media and print and electronic media should emphasize on vaccine, maintaining distance. Most importantly, avoiding crowding, gathering, rallies, elections. In my personal opinion, think we should not have any election for 1 year. Heavens will not fall. Elections are not required for economic activity. So also religious gatherings and functions. What is required for economic activity? Let us do that. But these are not required for electronic activity. Please avoid going to closed spaces where there are crowds. Let us be a little more disciplined and go in a regulated way. I think markets should be regulated. Indoor markets, if they can come out like vegetable and fruit markets, it will be much better. Let us continue to prepare more beds and oxygen, because adults also who are not vaccinated will get affected in the third wave. So we have to continue what we are doing now. We have to prepare for children. We have to prepare beds, hospitals. We have to start asking questions. Do we have to open colleges and schools for those below 18 years? If we open, are we going to test all of them before they come to college? After they come to college or school, are we going to continue to test them periodically like they do in the Western world? When there is a surge, are we going to interrupt offline classes and go back to online? These are things we should start worrying now and planning, discussing, and come with consensus so that we can protect people. In summary, then, SARS-CoV-2 is a new pathogen. It is contagious, spreads quickly. You can get immunity only through infection or vaccination, and the road ahead is bumpy. So we'll have to be prepared for it. One appeal is, if vaccine is available, you're eligible, please go ahead and get vaccinated. Vaccination prevents disease and death. It may not prevent infection. So you have to continue your responsible behavior. Please pass on this knowledge to others. Thank you very much. One last slide. These are the new words all of us learned: mutation, quarantine, social distancing. Mutation is change in the virus. But virus should not mutate. We should mutate in our behavior. We should mutate in such a way we start wearing masks and avoiding crowds. We don't need quarantining and social distancing for people. We need quarantine and social distancing for rallies, for elections, for crowds. New normal is to continue like this for another year. WhatsApp and other social media have contributed to an infodemic where more than 60% of wrong information is being circulated. It has done good, but it has done equal harm. And please be positive, spread positive messages. Don't only think of deaths and burning bodies and burials. We will get over this, but we have to be responsible. Thank you very much.
Dr. Ravi, thank you very much. It was a really very, very exhilarating, talk covering, sticking to the title of the talk and in-depth covering about the variants of this particular virus and what causes these variants, and went into the little science of it. And happy that I'm sure all our students, especially biology students, would have been extremely happy to know about how these variants are produced, and also talking about various vaccines and how they are developed and how they impact or how they interact and neutralize the a disease process or the virus mediated negative influences in the human system. Talked about it, and we also covered in detail about the precautions that needs to be taken. We would like you to permit some time and answer questions. There are some questions pouring now. And how much time do you have, Dr. Ravi? I initially planned.
I am at your service. You tell me, we will stop. Then I am available.
That's very nice of you, and I will try to ask. There could be some questions which might look like we have already asked this question. I've already explained just a few minutes back why questions are coming, but I will messages which are complimenting and appreciating your sharing your knowledge. There are some questions too. Be a little patient and answer all these questions. One of the question I'll start with is, why is virus only from variants only when it is spread among population? Let me understand why is virus only? Okay, I'm not very clear about it, but something to do with variants and its spread in the population. Why? Probably talking about differences in the transmission rates, I guess, about variants. Do you want to talk about it a bit? You have covered it, but maybe you can quickly recap.
Yeah, I think from what I understand is implication of transmission for variants. I think that that's what I tried to give examples that when the remember the fundamental rule in biology, in evolution especially, survival of the fittest. We know that. All the other variants which are not better than the existing ones will die down. It's a race between SARS-CoV-2 viruses. Somasekhar will always beat me because he's been a much better athlete than me. Viruses are also like that. So the one that fits better to receptors, attaches quickly, more firmly, is the one that will survive. So variants of concern, the 5 ones, all of them have better fitness for human cells. So they will, until a new variant comes, then Somasekhar, then that virus will reign the world for some time. So this is the rule in biology. So it continuously evolving, and the fittest ones will survive.
Thank you. Here is another question, probably which you did not touch about, is, sir, can you explain about white fungus and black fungus infections associated with COVID infections among populations? That is the question. What is the relationship? Why we are looking at it?
Both white and black are fungus. Both, both white and black are fungi. Black fungus is a misnomer. It does not mean that the fungus looks black. Somebody gave it a wrong name thinking it's Aspergillus niger. It is not Aspergillus niger. It is mucor. Mucor are fungi which, if you leave bread in a room for a couple of days, you will see something growing on the bread. All of us would have seen it. That is mucor. So this fungus, fungal infections occur in people who are immunosuppressed and who are diabetic. Even before COVID came, we have been seeing this fungal infections in severe diabetics at NIMHANS, where the thing spreads to the eyeball, and from there it enters the brain. In second wave of COVID, large number of people have got infected and they have received steroid therapy, and those among them who are diabetics are the ones who are developing this infection. If detected early, early signs are headache, drooping of the eyelid, pain in the cheek, pain behind the eye. They seek help at that stage, then you can give amphotericin B, which is actually a lipophilic amphotericin B because it has to be delivered into the cell. So it's liposomal amphotericin B. So that treatment, if you give, you can save patients. Sometimes the fungus is extensive, surgeons have to operate and remove the bulk of the fungus. Maybe eye surgeon, maybe ENT surgeon, maybe neurosurgeon. So this is being seen more post-COVID. When you have acute COVID, you will not get it. When you recover from COVID, at 2 weeks later is when you will see fungal infections. I hope I answered the question.
Yeah. So is it because of the compromised immune?
Exactly. And people think, that you get it like this. Actually, evidence very clearly says these fungi are already present in our nose. They get the opportunity to cause infection. People are saying from the ventilator you are getting, people are steaming and they are getting. Actually, data is very clear. People who have done none of these only compromised immune status. It is a ubiquitous fungus. It is found all over the universe. It is there in your nose and my nose also.
Okay. Thank you for that clarification. We have a question from Dr. Reena Shyam, who is a faculty in our college. Sir, how long does immunity last after COVID-19 vaccination? I think you did explain. Could you quickly, maybe a one line?
Now, as 9 months it has lasted. The first trial participants received vaccine 9 months ago. And we also know that neutralizing antibody levels are still there in them, and memory cells have been induced in many vaccines. So it will last 1 year.
Okay. A friend
Maybe beyond.
Okay. Thank you. Mr. Sunil Kumar asks, considering that vaccination of large percentage of Indians will take several months, as explained by you, how severe a third wave we can expect is the question. Dr. Ravi?
Actually, I'm starting a company and charge Sunil Kumar for it separately. That's the humor side of it. Okay, we have to expect that it will be as bad as the third wave. Because last time we did not expect, we landed in trouble. Let's make preparations. But how big the third wave will be will depend on two factors. One is that.
Okay.
I think I lost my connection. Oh no, it's there. Okay, right. So number of people that will immunize is one factor. Second, in our behavior of masking, social distancing, avoiding crowds and rallies. And the third, most important, is what if a new variant emerges that spreads even faster than the current one and may spread to vaccinated people? We do not know. So best is assume it is going to be to the same level as the second wave, because remember, half of the country, we are at 18 crores now. We have immunized 18 crores. With great difficulty, we may immunize 60 crore people still, or 80 crore people. Still, there will be 60 crore people left. So let us accept it is going to be a problem and prepare for
it. Well, Sunil Kumar also thinks since you are addressing students, probably he feels it's more a suggestion than a question. Maybe they would get impacted, influenced by your talk, and not only they get vaccinated when their turn comes, they will also convince their elder lot in their house to get vaccinated. That way, maybe this like movement of getting maximum number of people, population vaccinated would, that is an observation.
Exactly. I used to do this. I used to do this during the HIV days, saying that I think about how much, let me have a look. 456 people have attended this session. Each one of them should get vaccinated and make sure that, and when they get people vaccinated, get 2 people vaccinated. That moment you should start. It is a wonderful suggestion, and make sure that all students wear masks. Let Indian Academy group of institutions be an example that they are all masked students. Masked only physically, but not mentally. They should be bright mentally, but physically their nose and mouth should be covered. So I think this is a movement that has to come from us. Expecting government will solve problems is foolish. This is our problem. We have to do our. The responsibility of government is to provide beds, to provide oxygen, to provide vaccines, make sure it is available. Our responsibility is to vaccinate and everybody wear a mask.
Thank you very much for that, because when it comes from horse mouth, probably our students will take it seriously. That is one of the huge challenges we have, all principals, faculty, in making sure students converge into COVID appropriate behavior. I hope your guidance will help them. Thank you very much. I have a participant from Virginia Commonwealth University congratulating you and appreciating your effort. He has no question though, Dr. Karnam. And a faculty here, Dr. Amruta Roy, again, thanking you for your enlightening session and asking a question regarding vaccination. If a person is fully vaccinated and later that a new and improved vaccine becomes available in the market, is it advisable to receive the second improved vaccine as well? Is the question.
Very, very common question people ask me. And also people ask me, can I take a booster? See, this is, I'm not going to be prescriptive, but I'll tell you what I will do if it is acceptable. First, get 2 doses of whatever is available. All of them protect against disease and death. Next, please ensure that everybody around you has got the vaccine. After that, we will get greedy and say, I want 1 more dose, I want a better vaccine. Right now, we have to make sure that as many people get the vaccine so that all of us are safe. If I go on taking Covishield followed by Covaccine followed by Pfizer and Moderna, I'm depriving vaccines for much more eligible people out there. So let's be responsible.
Yeah, I'm sure that will go down well. People to be little concerned about others' requirements before we getting too much. Here is another question from Dr. Karnam, Srinivasa Karnam, from asking about, does the reinfection depend on the neutralizing antibody titer value, given the fact that antibody titer varies? Or is it reinfection due to low antibody titer in the vaccinated person? Hope you understood the question. If you want, I can.
I didn't hear that, Dr. Somasekhar. I momentarily lost connection.
Okay, let me repeat that.
Yeah.
Does the reinfection depend on the neutralizing antibody titer value, given the fact that antibody titer varies?
Very true. It depends on the neutralizing antibody titer. That is the surrogate marker for protection. It also depends on variants. I may have very good neutralizing antibody, like I showed you some data with vaccines. I may have fantastic antibodies, high titers to Wuhan virus, but it may or may not protect against the Maharashtra mutant to the same extent, or one mutant that is going to come tomorrow or day after. So reinfection will depend on several things. One is your titer, 2 is the emergence of new variants, and 3, most importantly, variants, no variants, none of them penetrate mask better than the other. If you wear a mask, if you follow simple things, there is no need to get reinfected.
Okay, thank you. And here is another question. Are the mutations in the spike proteins making the virus more severe, or the virus is programmed to behave so? I'm not sure I understood it, but...
Yeah. Okay. Can you read again? I try to make sense of it.
Are the mutations in the spike proteins making the virus more severe?
Yeah. Okay, I understood. He's asking the question, the person is asking whether the implication of spike protein mutations on virulence of the virus. Well, as I showed you, it is data evolving. There are some data with the Brazilian and the UK variant saying that yes, it has made the virus more virulent in certain populations. As far as the Indian double mutant is concerned, hamster data says it is more virulent compared to the parent Wuhan virus. But we don't have data from humans. It's going to be very difficult to collect that kind of data, looking at people who are infected with Wuhan virus and then those with virulent. Yes, short answer to to a long explanation is mutations in spike protein can alter virulence of the virus.
Okay. Here is a question from a BBA student for a change from away from science backgrounds. Deepa asks, a person who is allergic to allopathic medicine, some of the antibiotics, let us say, can they take vaccine?
Yes, I have personally counseled about 20 of my doctor colleagues and other friends who have severe allergies. So my advice is very practical, and that is based on clinical medicine principles. Any drug that you inject is supposed to be first given as a test dose. It's a different matter, people don't do it. You must have heard of penicillin allergy. Anybody who gets penicillin injection, first they inject a small test dose in the arm, zero point 0.1 ml intradermally. Wait for half an hour. If the person has rash, tingling sensation, increased breathing, that means he's allergic. You have to treat him. That's how in vaccination in COVID, we ask them to wait for half an hour. So I would say if known allergies are there, take a test dose, wait there 2 to 3 hours. If you don't have any reaction, go ahead with the full dose. Many of my friends have done and they've been very happy with this.
Yeah. Here is a question from a student who asks, which kind of vaccines are good for mutants and double mutants? Killed vaccine like Covaxin or subunit vaccines having spike proteins?
Very practical question.
Talked about it. Yeah.
Very practical question. Theoretically, and some evidence points out that whole virus vaccines are better. I mean, it's common sense. In whole virus vaccines, you are giving the opportunity to the immune system to respond to all its proteins. Whereas in subunit vaccines, only spike protein. The moment it mutates, then you have to change your vaccine. Whereas whole virus vaccines, I always give an example. If you can't catch the lion by its face, you should catch it by its tail. And that co-vaccine can do.
Yeah. Here is a question from a faculty from our PU college, Mr. Uddhav Khatri. Thank you, sir. Are the vaccines for children different from the vaccines of adults?
Very important question. The vaccines are not different. Currently, Pfizer has been cleared for use between 12 and 18 in America. Many vaccines are undergoing trials in children. The vaccine is the same. The vaccine is the same. The dosage may vary depending on the vaccine, and for that you need trials. Schedules will remain the same. The regulators did not clear for children and pregnant women because they were not included in the trials. Remember, we ran an emergency trial. That time, we did not include children and pregnant women. But believe me, they will also be safe there, as far as I know, because these vaccines are all safe. But unless data is produced, it will not be clear. Bharat Biotech has been cleared for children's trial between 2 to 18 years, 3 days back. In 3 months, they will get hopefully clearance after they produce the safety data in children.
Yeah, thank you. I think quite a few people have asked questions. There are more to follow. Let me quickly get them to you. There is a question about a political kind of question. I don't know how we are going to answer. Is this a kind of deliberate, indirect, or direct attack on some countries? Like countries by China, something like that. So is it originating from? I think that there's a lot of news about
going on and off. It started as a lab-generated virus. Then there were papers published in Science and Nature saying, no, it has 99% similarity to bat and pangolin virus. So it is not man-made virus. Again, now we are seeing in social media a lot of debate saying that we will never know. The fact is, there was a virology institute in Wuhan and they did work on coronavirus. Whether it's a deliberate leak, whether it's an accidental leak, or whether it is because in the wet markets, if you look at earlier pandemics, the way they emerged because human beings, animals, and birds crowd together. That is why now we talk about a new area of collaboration known as One Health. Future pandemics will evolve in areas where there is human, animal, and bird crowding. Exchange of virus will happen. Also in areas where there is human animal conflict, edge of deforestation. So many examples. I don't think that's a seminar by itself. Sometime later we'll do on how human beings invite trouble for themselves. Viruses are wonderful, innocent organisms. I've become an advocate after 40 years of work. They live in their niche. You go and you cause and call them out, invite trouble. I will not answer the question whether it is man-made or not. I don't think this is a parallel argument. It will keep going. You will not find evidence.
Yeah, well, we will. There are quite a few questions on that line. I'll avoid them since you have answered it. Here is one lighter vein question, sir. How long a reusable cloth mask can be worn at a stretch?
The way masks are used in Bangalore and including some of my friends, they are used eternally. No mask is eternal. Let me give you 3 points about mask. Mask is effective for 6 to 8 hours. Beyond that, the moisture in your breath will make the mask wet. And when the mask is wet, water molecules act as transport of virus from outside to inside. Disposable masks should be disposed, not dried and reused. Every day you should wear a different mask. Ideally, when you are in a ward treating patients, every 6 to 2 hours, you have to change your PPE and you have to change your mask. If it is cloth mask, it should be triple layered. It should be washed. Soap and water kills the virus. It should be sun dried and ironed with an iron box and worn the next day. But I have seen many people wearing the same mask at least for a week or 10 days. Fantastic recipe for disaster to invite infection.
Thank you. I remember you telling people in another meet, we use, if possible, different towels and try to.
Yeah. Dupatta is not a mask. A towel is not a mask. When you see a police, her kerchief is not a mask. Please.
Yeah. Himangi Bave asks a question. Sir, thank you so much for your time. I am 30-year-old and a mother. I tested positive in the month of April and then got it negative as well. As you have mentioned, vaccine we can get done after 3 months, but what about if I want to donate plasma? Am I eligible to donate blood plasma to needy? While answering this question, Dr. Ravi, I would like also to touch upon the relevance and validity of convalescent plasma therapy for COVID patients.
Okay. Let me say that yes, you are eligible to donate plasma if you are not a lactating mother. I thought she said she's a mother, right?
Yeah.
If you're a lactating mother, lactating women are not permitted to donate. Okay. So that is something that she has to remember. Number 2, you should take a vaccine 90 days after your PCR test came positive, because you will have to boost your immune system with a vaccine so that your memory of the immune system lasts at least a year long. Now, about plasma therapy is again flip-flop. The jury is still out there. There are trials which have said it's useful. There are trials which have said it's useless. There's an NEJM paper said, yes, it is useful, but in a very limited setting of early infection. But remember, plasma therapy has to be very regulated. That means the plasma you're giving to a recipient has to be high titer plasma. If you mix low titer and high titer plasma, antibodies will get diluted. The funda is that you're trying to neutralize the virus before it goes down to the lung and produces a cytokine storm. We all know COVID disease is not because of the virus, but it's because of the aggressive way your immune system responds to the virus. Before that starts, and that window of first 3 to 4 days or 5 days, plasma therapy is beneficial.
Yeah. Thank you for that response, for clarification. Dr. Malayaris Pandian, faculty here, asks, in the present situation, it is believed COVID does not spread through surface transmission. Then why wash hands, fruits, vegetables, and groceries before being used?
Cleanliness does not do any damage. Please do not resist anything that suggests to wash hands and wash things. You will not get, today it is COVID, but you will not get another dozen infections by doing this. So look at it positively, number one. Number two, surface transmission is not completely ruled out. It depends on I walking into your room, sneezing with COVID and going away, you come next moment, droplets will still be wet, you touch them, you put your hand, you'll still get infected. But the probability of that is less compared to human transmission. But it is definitely there. So please, do you want to take a chance? It's up to you. As I told you, I will not be prescriptive. You don't want to wash your hands? It's up to you.
Yeah. Here is one question which I think is to some extent contributing and choking the resources, hospitalization resources, making the whole issue very much more complex. Sahana is asking, a student of microbiology in our college, when does a person actually need hospitalization and treatment?
Because the subject was variants and vaccines, I did not go into my usual clinical symptom. There are some questions I saw in the chat box on about symptoms, and let me tell you, if you look at the natural history, we have learnt it very well over the last 1 year. Symptoms start with fever, cold, cough, myalgia, headache, loss of smell, some people diarrhea, loss of taste. These are the common symptoms. And even many people do not have symptoms. But after virus enters, usually within 4 to 5 days, people get symptoms if they get symptoms. Once they get symptoms, 4th or 5th day is when a proportion of people will start going downhill. So the second week of illness of infection is very crucial. Between 5th and 7th day, 5th and 12th day, people have to be monitored. In second wave, the biggest beating that was taken is lack of monitoring of people in home. Even asymptomatic people, between the 5th and the 12th day, they can deteriorate. If the virus has gone to the lung and induced a very aggressive immune response, that's when your oxygen starts falling. So pulse oximetry is paramount, and taking a notebook and making readings every few hours when you're awake is important. Very early indicator of deterioration is 6-minute walk test. Take pulse oximeter reading, walk briskly for 6 minutes, repeat the reading. The fall in oxygen saturation should not be more than 5%. If you're 97, you can come to 92. But if you come to 90, that means you are developing a very aggressive pneumonia. You need to get into a hospital. So all investigations are very useful in the second week. I turned positive. I don't panic and go and take a CT scan and D-dimer and ferritin and that and this. If I do, everything will be fine on the first, second day. What is more important is to do it. And there is a window for everything in COVID, including testing and positivity. So I think that's very important.
Yeah. Thank you very much. There are a lot of questions related to comorbidity issues. If I have I'm diabetic or hypertension, why people with comorbidities are, I'm trying to put them together and ask it.
That's a bunch. I can give you a summary of comorbid. See, your immune system works best when you are healthy and you don't have other diseases. When you have other diseases, your immune system is compromised. And you also get complications in COVID. Anybody who has a vascular disease like hypertension, kidney and all that, clotting becomes more troublesome for them. A healthy person is able to remove. Clotting is one of the features that occurs in COVID. Moderate and severe COVID, the blood vessel lining is damaged. Whenever a blood vessel lining is damaged, platelets come there, cells come there, and a small clot is formed. And clotting is one of the post-COVID complications. You see heart attacks, you see strokes, you see kidney injury, and you also see pancreatitis. So if your immune system is optimal when you are healthy, if you have diabetes, you have hypertension, kidney disease, cancer, HIV, your immune system is compromised. And when it is compromised, you get an aberrant response, and that's what gives you serious disease.
Yeah. Here is another question asking about does the rate of mutation increase when there is resistance to the virus in the vaccinated person?
Theoretically, yes, vaccination will drive more mutations. That is the nature of vaccinations. But then the benefits of vaccination are far too much to worry that no, we will not vaccinate people because we will drive mutations. As I told you, mutation will continue to occur, but fittest among them will be very, very few that emerge. Most of the mutants fall by the wayside and they become useless or they are neutralized.
Basically, a race between
Yes.
virus.
Yes, between viruses.
Yeah. Why do Grace Peniel asks, why do they give steroids for COVID affected
I told
people?
you, COVID disease, the lung disease in COVID, the severity in COVID that we see is because of an aggressive immune response. Steroids suppress aggressive immune responses. That's how we give steroids.
Yeah. Thank you. Rachana Tumula asks you a question. Could you please say?
I saw the question. I thought I'll answer it last.
Could you please say more about general health measure that we can take to improve our immunity in ways that can aid the natural development of antibodies and help improve our response to the virus?
I will give you what scientific facts, proven scientific facts are there. Proven scientific facts are vitamins are very important for a good immune response, especially vitamin D is an immunomodulator. And many people, unlike our childhood, Dr. Somasekhar, most of the youngsters today are not exposed to sunlight, sedentary lives. So they don't make enough vitamin D. Gross deficiency has been noticed by many surveys. So vitamin D is very important. Number two, vitamin B complex is also important along with zinc. Zinc is a very important catalytic agent in several immune reactions. So zinc should be required. And for a body to have a good immune system, nutritious diet is important. And lot of fruits, vegetables are important. So these are very, very important. If you go to natural foods used in our traditional systems of medicine for many years, please take it. It doesn't do harm. It does. I don't know if they're immune boosters. There is no data. But if it makes you feel good, please take it. Anything that makes you feel good is important because the whole science of psycho-neuroimmunology tells you if you are happy, your brain is happy, it drives the right kind of hormones and then the right kind of immune response. Above all, good quality sleep is very important for a good immune system. And people feel happy when exercising because there's a lot of release of endorphins. That keeps your mood good. That is very important. So exercise, definitely. And we know from yoga, there are studies that tell you that yoga is a very good potentiator of good immune response. Meditation, yoga, there are papers in psychoneuroimmunology journals that say they promote immune response. So these are the things I would say are very important.
All good things that needs to be. Yeah, there were many people who had asked about yoga, meditation. Before I could come to that, you have answered. Thank you very much for that. One of our senior faculty members asks, after taking 2 doses, for how many years we will be safe? I think you answered it, but maybe one more time to.
Okay. As of now, 9 months, but I can tell you 1 year. It all depends on whether mutants are going to arise and spread faster. So it looks like the way in the last 4-5 months, the number of mutants that have come, although corona is not a virus as as labile as influenza, but I'm getting a little worried whether we may have to have annual boosters with a new cocktail like we do for influenza. I don't know.
It's an unfolding situation,
Yes.
so we may have to wait. And
we do not know. Yeah.
Yeah. Hrideshwar Shiv Prasad asks, sir, how is the working of the nasal vaccination and how effective will it be in the long run?
As of now, it looks exceedingly promising, and if it is, I pray that it is effective in phase 3 trials. Then it will be a game changer. It will be a game changer because this is first time we are neutralizing virus at the point of entry. You build a local immune response in the nasal mucosa and in the oral mucosa. So when virus comes, immediately it is neutralized and you prevent infection. That has happened in animals in phase preclinical stage. And there are 4 companies making nasal vaccines in the world. One Australian company vaccine is in a very advanced stage. Our own Bharat Biotech is now going to phase 1 trials. So let us hope, we live on hope, nasal vaccines will arrive and they will be vaccines that can cut down transmission and prevent infection.
See, this question is a bit, I think, although repetitive, I think it deserves to be answered with a little bit more emphasis. Sir, why people are dying after getting vaccinated? You talked about it, but that should, I think there is a amount of skepticism which I would like to be eliminated. So maybe you should give some attention.
I'll answer it in the social aspect, and I'll answer the scientific aspect. People get infected after vaccination. Every one of us in this 397 people on the show right now knows somebody who's vaccinated and got infected. Okay?
Yeah.
The infection, it does not prevent. But after getting infected, one has to really monitor oneself. I told you the critical period is 5th day to 7th day. If people are not monitored well, and if they have comorbidities, then it's better that they should get into a hospital so that they are under supervision. And if they deteriorate, immediate oxygen and drugs are available. So that is something I want to say. It's unfortunate. One of the leading doctors whom I knew, Dr. K.K. Agarwal, died the other day after 2 doses of vaccine. Very, very unfortunate. So we have to, from our side, be careful. Even if you're vaccinated, you get infected, we should monitor or get into a hospital. That's number 1. Number 2, there is too much of social media attention on the deaths. I'm not saying it should not be there, but actually, where is hard data? 18 crore people have been vaccinated in this country. In that, at least 8 crore people have received 2 doses. How many people have died in those 8 crore people after vaccination? Do we have data? We do not have data. Yes, it is unfortunate for an individual that he dies after vaccination, but that does not mean vaccination is useless. Please don't go. The benefits of vaccination far outweigh the risks, if at all. Please.
That has been very clearly told. But in one of your slides, Dr. Ravi, what I have noticed is post first dose of vaccination, infection rate was 0.2, something like that. And then after second dose, it was a bit more.
Yeah, but
I mean, still in subdecimal points, but
It does not mean that after second dose, your risk of dying is more. It is just that numbers are different in the two groups, post first dose and post second dose.
And you're dealing with small numbers and evolving situation.
Yeah, exactly.
I hope we have stretched your time by 25 minutes. I'll take 5 more minutes to close it. Is it okay, Dr. Ravi?
Fine, no problem.
Yeah, okay. Banita Behera asks, sir, already 20 days I tested COVID positive. I have taken medications as per the doctor, but still I am weak. Cold is also there. So kindly suggest what should I do? Even for my husband, it is 15 days over, but he has cold. I think why I'm asking this is many people do complain post even 3 months, 6 months after they have recovered, feeling weak and having, symptoms are lingering, like impact of.
Let me answer this question. The answer to the question is majority of the people recover within 14 days.
Yeah.
So a quarter of them or 20% of them will have symptoms prolonging for a long time. Having cold, one thing very important is to keep a notebook and then every day write down whether your symptom has worsened, whether it is better or whether it is same. Cold. Is my cold more today than yesterday? Is it improved or is it the same? Be honest to yourself. That is very important even for doctors to give you the appropriate treatment. I run a WhatsApp OPD in the mornings between 7 to 9:30 for about 40 people, my close friends and relatives. This is what I ask them. How are you feeling? Is it better than yesterday? It is the same. These are things that you have to monitor. But having cold and cough, I have seen in some people it can go on for 3 weeks. Please don't worry. If it is worsening, please go and see a doctor.
Yeah.
Monitor when you have symptoms, please monitor your temperature and your oxygen till you become afebrile or till the temperature comes down. People can develop complications later. I'm not scaring you, but since you are at home, it's your responsibility to find out whether you're worsening or improving. But don't worry, it will settle down. Some people take 4 weeks to settle down. And Dr. Somasekhar, your question, COVID is not merely a respiratory disease, it's a multi-system disease. In the fight against COVID inside our body, we produce so many cytokines and inflammatory markers. So tiredness, all these will last a long time and people will take time to recover, especially those who have had moderate and severe illness. Asymptomatics within a week or 10 days, they're out, they're absolutely fine.
Yeah. There is one question which I thought you should answer. Shubha Badran, a faculty in our college, asks, if someone has taken a dose of Covaxin at first dose, but the second dose of the same is unavailable, what we are supposed to do is, can we take another vaccine?
To wait and take the same vaccine. Again, remember, when regulators approved, they approved it for 2 doses of the same vaccine. Many people ask me, no, why can't I should have choice. This is not about democratic rights. It is about science. 2 vaccines of the same manufacturer, you get protection is what he guaranteed. Right now, interchangeability trials are going on. For example, Pfizer and other viral vectored vaccines, people are doing trials. Sputnik is tying up with Astrazeneca because both are adenovirus vaccines. So unless results come out from bridge trials of interchanging one with another, till then you will not be permitted to take.
Yeah.
Please don't experiment. I know
a good suggestion.
It's disturbing. Don't worry. If it is delayed by a couple of weeks, any vaccine, it's not that the immune system exactly at the end of 3 months forgets what Covishield is. No. The longer the interval, in fact, you boost the immune system much better. Memory cells will kick in. But why are we not giving every vaccine so long? Because it's not practical. There'll be dropouts, and during the long interval, you can get infected. So it's a titration to give you the best benefits. In case it is not available for a couple of weeks, don't get worried. You don't have to restart your immunization schedule. You can take the same vaccine.
Here is one long question, looks like very technical. Thank you, sir, for more information about COVID-19. Actually, adenovirus viral vector can acquire E1 gene during production in HEK293 cells. My question here is, adenovirus restores its ability to replicate? If yes, then what is an acceptable limit of replicating virus particles per vaccine dose? Is a biochemistry faculty.
I know.
So,
actually, I would encourage him to go to the FDA website. FDA has very clearly defined the permissible amount of replicable virus that should be there in a formulation. Now, this was a question that came up before the Sputnik board because Brazil, for non-scientific reasons, because the government changed and some other things, rejected Sputnik vaccine, saying that it does not meet these FDA standards of so many particles, so much of replication is permitted. Then they actually showed us the data saying that it is much below the limits set by FDA. So it is a very valid question. It's a very valid concern people have. What if, see, right now the adenoviral vector vaccines are designed in such a way that they enter, they infect a cell, deliver the gene, and then there is no replication because E1 protein is lacking in that. So it's a suicidal kind of bomber terrorist attack. It goes inside, does, and then it dies. But yes, there's a very, very, very remote possibility of it reverting. So that possibility will exist with vector vaccines. So I'm being very honest. There is no guarantee for anything in life. To live is to risk dying, as a philosopher. As long as you're alive, you have the risk of dying.
Yeah, I was expecting that quote from you. Dr. Srinivas Rao asks, his question is, if a person is affected with COVID, he or she, can they take alternative medicine system like Ayurveda medicine, and is there any Ayurveda medicine available? I think to just add to this question, maybe it is relevant. I just saw yesterday in a news clipping where the people, I think some 10,000 people were in front of some miracle medicine in Nellore, Andhra Pradesh. I mean, the crowd itself was scary to see people shoulder to shoulder. Maybe combined, maybe you could address this.
You see, I want to give a very open answer. Traditional medicine, alternative systems of medicine have been around for centuries. But the problem in understanding their efficacy is there are no rigorous clinical trials the way it is done for allopathic medicines. Probably they are good, I don't know. But in the absence of evidence, to rely only on that may not be good. Even if you do, please monitor your pulse oximeter. If you deteriorate, forget all traditional medicines, go to a hospital, get
Run to the
oxygen,
hospital.
get it. Yeah, but yeah, see, if it makes you feel good and happy, take it. It's not going to interfere with allopathy, all the medicines that are available now. But remember, if you do deteriorate in your oxygen saturation, you have no other choice but to run to allopathy right now.
Yeah. In this context, I want to ask you some of the really renowned, famous people with medical background and former, leaders in medicine and still visiting professors in some of the big universities in UK, going ahead and talking in open forums that vaccination is light of vaccination and modern allopathic approaches. I know there is nothing we can do about it, but
Yes.
it's frustrating to see these things. There's one quick question before we close. If a person is already infected and came out of infection, or at some point, should they go for both doses, or one dose is enough? Whenever, after 6 months, 3 months, whenever they can.
The answer is we do not know. But having said that, let me tell you, Johnson vaccine is going to get I think it has recently got approval for single dose. The whole schedule is single dose, but with a rider that the protection is only this percentage. That the reason why it is being approved is that you can give large number of people you can vaccinate when it's single dose, and you can bring down the burden of disease and death. I would say at this point of time, there is no evidence, compulsive evidence to say that one dose is enough. So, as I said, immune memory wanes after 3 to 5 months, after which you are definitely susceptible, not as much as a naive person, but you still have the chance of getting infection. So go ahead and take 2 doses. Why do you want to experiment?
Dr. Ravi, thank you really very much for the time that you have given and patiently answered all the questions. But I want to tell you there are probably another 100 questions which I am not going to go into because I've already taken enough of your time, and I would probably collate and send across to you at some point. Maybe if you have some time, maybe you could respond.
Maybe somebody can read out. You can take a half an hour phone call and I can answer them, because
I'll share your email if you permit me with participants. They could probably directly send across a question. I know you are very busy these days, chairing so many meetings and attending so many sessions. So whenever you get time, maybe you could answer them, and that way they would find it, like they would not be disappointed that Somasekhar did not read their question to you. And thank you really very much for the time and the effort that you have put in, and passionately answered all the questions. I also would like to thank all our principals and our COO, Dr. Srinidhi, for helping me to coordinate and get this thing organized. I would now request Professor Rajarajan, head of our microbiology department, to formally propose vote of thanks and close this session. Professor Rajarajan, over to you. Are you there?
Yes, unmute.
Yeah. Rajarajan, you have to unmute.
Thank you, sir. Your talk was really, from a faculty point of view, it was mind-blowing, because we could grasp quite a lot many which probably a textbook or even the Google cannot teach us. It was really mind-blowing. And with that, sir, it is certainly my privilege to have been asked to propose a vote of thanks on this occasion of such an enlightening and wonderful event. I, Professor Rajarajan, on behalf of the Department of Microbiology, Indian Academy Degree College, Autonomous, and the entire fraternity of Indian Academy Group of Institutions, here to extend a very hearty gratitude to our eminent resource person, Dr.V.RAVI, for gracing this occasion and sharing with us his very valuable thoughts, which should have enlightened many minds and dispel lots of myths about the do's and don'ts in the present scenario. Sir, I'm sure your insights today certainly threw enough light on the nature of virus to have emerged stronger than earlier, or the precautionary measures to abide with, or the road ahead, with your really inspiring address. Thank you so much, sir, for your time and patiently taking up the questions and answering them.
Thank you. Thank you so much. I always learn when I do a session. Thank you.
I'm indeed elated to express my gratitude to our benevolent leader and educationist par excellence and a figure of inspiration to the entire Indian Academy family for organizing this webinar, our Honorable Chairman, Dr. T. Somasekhar, so that our students, faculty, non-teaching faculty, and their dear and near ones are enlightened. Thank you, sir. I'm sure the talk would have immensely benefited everyone who were a part of this session.
Especially our students, a large number of them have been participating and listening to Dr. Ravi. It was primarily planned for them, and I hope they're happy to, some of the questions they have raised, we have put across to Dr. Ravi. And Professor Rajarajan, you need to know from Dr. Ravi, maybe you can communicate to him through email.
Yes,
that Bharat Biotech is setting up. I was told maybe Dr. Ravi can just mention a couple of sentences about it, and which is a huge opportunity for life science students in
a huge
Bangalore.
Production facility being getting ready in Malur, very close to Bangalore. Malur is in Kolar district, very close to Bangalore, 1 hour drive. So I was thinking that, maybe that students of microbiology and biotechnology, it will be nice for them to have a visit or, have placements after they finish their course, contribute to vaccine production. Because human resource is one key challenge we have faced, whether it's in treatment of patients, diagnostics, vaccine production, any of these. So I think your institution probably should get in touch with Dr. Krishna Ella.
Certainly, sir. Certainly, sir.
Well, thank you very much, Dr. Ravi. Really appreciate your time and effort. Once again, we will close the meeting with your permission.
Yes.
Thank you. Goodbye.